Hello everybody, today we're going to talk about oxygen deficient zones. I'll explain what an oxygen deficient zone is in a little bit, but first I want to start just by highlighting that a big theme of this lecture is that the carbon cycle and the nitrogen cycle, whether we're talking about the ocean or land or lakes or wastewater treatment plants, are connected. Can't really understand the carbon cycle without the nitrogen cycle and vice versa. And to start explaining what I mean by that, here is our cartoon of the ocean. Once again, we've got our phytoplankton who are autotrophic and fixing carbon dioxide, releasing oxygen as their byproduct. Animals and other organisms are pumping carbon down to the seafloor as part of the biological carbon pump. And then heterotrophic bacteria are consuming this organic matter while they breathe oxygen. And they release carbon dioxide as their byproduct. Okay, so let's review. This is what we've talked about before. We've also talked about how if there is more organic matter in a given environment, that is a depth in the ocean or a location in the ocean or on land, a particular patch of soil or a particular lake or a river, if you have more organic matter, then there is oxygen in that environment, then anoxia will result. You will use up all of the oxygen because of all of that organic matter. And we've talked about how when you run out of oxygen, organisms will then move on to other electron acceptors. And that opens up the whole world of anaerobic metabolism, which is very diverse because there are lots of electron acceptors that are not oxygen. Nitrate is usually the first one that the microbial community will go to once oxygen is gone. Things that use nitrate as their electron acceptor are often denitrifiers. That is, they convert that nitrate into nitrogen gas, the N2 molecule. After the nitrate is gone, then the community might use things like sulfate. Sulfate-reducing organisms often produce sulfides and other smelly sulfur molecules. And CO2 is the electron acceptor that is used by methanogens. And we've talked about how methanogens are special and that their electron acceptor is also their carbon source, which is one reason why people have argued that methanogenesis may have been one of the earliest metabolisms to ever evolve, because their electron acceptor is also their carbon source, so those two things are merged in methanogenesis. Okay, so this is all review, setting the stage for what we're going to talk about today. And we're going to talk a lot about denitrification and other members of the nitrogen cycle and how they relate to the carbon cycle. So once again, denitrification is the respiration of organic matter, that is the consumption of organic matter with nitrate or nitrite. Nitrate and nitrite behave very similarly in all of the examples we're going to talk about today. And the product is N2 gas. And this is called a nitrogen removal process in the context of many ecosystems because N2 gas is a gas, it escapes. So because it's a gas, it will be removed from the marine ecosystem. And in this way, denitrification is actually removing nitrogen atoms from the ocean and releasing them back into the atmosphere. Now remember, this is different than nitrification, which we've talked about before. Nitrification is the recycling of ammonia back to nitrate or nitrite. This is done by completely different organisms than those that do denitrification, and they're actually different from each other in multiple ways. So denitrification consumes nitrate. Nitrification produces nitrate, so they have completely opposite relationships to nitrate. Denitrification is heterotrophic. Denitrification is consuming organic matter. Nitrification is autotrophic. Last time we talked about how the nitrifying archaea that carry out nitrification in the ocean are autotrophic. They are also fixing carbon dioxide as part of their metabolism. Denitrification is, of course, anaerobic. That's by definition. It is using nitrate or nitrite as its electron acceptor, so it is by definition anaerobic. Nitrification is aerobic. The ammonia is recycled to nitrite or nitrate with oxygen as its electron acceptor. So they are the opposites of each other in at least these three different ways, which is really important to keep in mind. This figure is a good summary of many aspects of the nitrogen cycle. And one thing that it is highlighting is how nitrification is the main aerobic portion of the nitrogen cycle that occurs in oxic environments, and denitrification is the main anaerobic part of the nitrogen cycle that occurs in anoxic environments. And pretty soon we're also going to talk about anomox. And nitrogen fixation, which we've mentioned before, is also anaerobic. One quick note about denitrification before we move on to tell more about our story of marine ecosystems, which is about the evolution of denitrification and the change of ocean conditions over time. So this graph is from a really short and entertaining article from 2007 that shows the expected, partly measured, partly modeled and predicted concentrations of various metals in the ocean over time, starting 4 billion years ago when the Earth first formed to the present on the right-hand side. And for example, we've talked about how iron is often limiting in most parts of the ocean. So this is where iron ranks among the metals in the ocean today. And copper, which is what we're going to focus on this slide, is even lower concentrations and is also very often limiting for many organisms and many communities and many environments, especially in the ocean. This is important because many enzymes require copper as a cofactor, just as many enzymes require iron as a cofactor. And in particular, one very key enzyme in denitrification, which is the final step that converts nitrous oxide into gas. This is the last step of denitrification, going all the way from nitrate to nitrite to nitric oxide to nitrous oxide to into gas. This last step, this enzyme, is nitrous oxide reductase. It requires 12 copper atoms in the catalytic center of a single enzyme. 12 copper atoms in one enzyme. That's a lot of copper for one enzyme, and therefore this step of reducing nitrous oxide to N2 is often subject to copper limitation. So when copper is limiting in the ocean, organisms that would normally be producing N2 can't, and they have to stop at the nitrous oxide step. And that is important because nitrous oxide is a really potent greenhouse gas. It is more effective than carbon dioxide at increasing the temperature of the atmosphere when it accumulates in the atmosphere. So that's important for understanding the nitrogen cycle and the carbon cycle today, and important for understanding global warming. The degree to which carbon is limiting in the ocean could be affecting the amount of nitrous oxide in our atmosphere. So that's an important thing to study, and we actually don't really understand it that well. And it's also interesting to think about, coming back to this figure, how things have changed over time in the ocean. And you'll notice in this figure, it's highlighting that in this period of time between roughly 1 to 2 billion years ago, which is the Proterozoic era, copper concentrations were at their minimum. This is because the ocean actually became sulfidic during this time. You see this dotted line is showing a spike in the concentration of hydrogen sulfide during the Proterozoic. So the whole ocean might have been super stinky and full of sulfide, and that would have precipitated out a lot of these metals. So copper sulfides are expected to have formed and precipitated out and landed on the seafloor, reducing the availability of dissolved aqueous copper in the surface of the ocean, potentially causing widespread global copper limitation, which may have prevented nitrous oxide from being reduced into gas globally, and that might have led to an accumulation of N2O in the atmosphere, perhaps causing a laughing gas atmosphere, is the title of this paper. Nitrous oxide being laughing gas, of course. And this could have big implications for understanding the climate at the time because, again, nitrous oxide is a good greenhouse gas, and so this would have affected the temperature of the Earth at the time. So I think that's a fun story, connecting all these different elements together into talking about a sulfidic ocean a billion years ago that was full of laughing gas. Okay, coming back to today. Again, we've been talking about how denitrification and nitrification are complete opposites of each other. They reverse each other. And now we're going to talk about anomox, which is another process. It plays an important role in the nitrogen cycle. It was only recently discovered, especially the organisms that conduct it were discovered only when I was in graduate school. And it's not exactly the opposite of denitrification. It has many similarities to denitrification, but it also has many differences. So let's talk about it. Anammox, again on the previous figure, we can see how anammox also produces into gas, so that's a big similarity to denitrification. It uses nitrite as its nitrogen source, as its electron acceptor, which is similar to denitrification. However, it skips these intermediates. It does not have a nitric or nitrous oxide intermediate. It goes straight to N2, and it uses ammonia as its electron donor. So instead of being heterotrophic and using organic matter as the electron donor for producing N2, it's actually using ammonia as its electron donor, similar to nitrification. So in some ways, you can almost think of it as a hybrid between denitrification and nitrification. It's producing N2 from nitrite, like denitrification, and it's consuming ammonia, like nitrification. So that occupies this kind of intermediate weird place in the nitrogen cycle. And the organisms that do it are also very weird. The only known organisms that can do anammox, at least in this way, are in the phylum Planktomycetes, which is a famously weird phylum. The organisms in this phylum are kind of loners. They have very different biology. In particular, they have lots of compartments inside their cells. When these compartments were first discovered, they were proposed as like missing links between bacteria and eukaryotes. I don't think that they're a missing link. I think they're just really interesting on their own. It's probably an independent evolution of compartmentalization inside a cell, which I think is perhaps even cooler than being a missing link between bacteria and eukaryotes. But anyway, one of the compartments that some of the species in this phylum have is an anammoxosome, which, as you might expect, is a compartment where they conduct anammox. Here is the molecular pathway, and the main thing I want to highlight here is that an intermediate in anammox, so here is nitrite and ammonia, as I said. These are the electron donors and electron acceptors for anammox. Producing N2 is the product, nitrogen gas, and the intermediate is N2H4, which is hydrazine. Hydrazine is jet fuel. So if you are producing jet fuel inside, or rocket fuel, that is not jet fuel, but rocket fuel. So if you are producing rocket fuel inside your cell as an intermediate, you probably want to contain it inside a compartment. So that is probably one reason why they have these anammoxosomes. And another thing about these anammoxosome is that the lipids that form the membrane around the anammoxosome have these very strange square-like lipids. So these are the lipids that make up the membrane of this anammoxosome compartment. And they have this square structure in their tails. And it's thought that the square structure forms really, really tight seals in the membrane around the compartment. And you probably want this really tight seal to keep in the hydrazine so that the hydrazine does not escape from the anammoxosome and then toxify the rest of the cell. So that's some really cool biology. There are lots of papers about anammox that you can learn if you want to learn more about the biology. That's a little taste of these weird organisms. And remember, it's important to remember that these weird organisms are the only ones that do this anammox process. They're the only ones that can produce into gas using nitrite and ammonia. Okay, so let's put anammox into our cartoon of the marine ecosystem and think about some consequences of to the ecosystem now that we understand how denitrification, nitrification, and anammox relate to each other. So remember, typically in a healthy marine ecosystem, you're going to have oxygen all the way down to the seafloor if you're not producing a dead zone somewhere. That oxygen will be consumed within the first few centimeters usually in the seafloor, in the sediment, the mud at the bottom of the ocean. And then once the oxygen is consumed, then you get denitrification and the other anaerobic pathways. But when you have enough organic matter and you have other conditions that allow the complete consumption of oxygen still in the water column, so the oxygen has already been depleted in the water before you even get all the way to the bottom, that is when you have the potential of having something that's called an oxygen deficient zone. So when oxygen is rapidly consumed in the surface water, then you get denitrification, NMOX, occurring in the water column, whereas normally you would only get such anaerobic pathways occurring in the sediments. So let's talk about these oxygen deficient zones. Sometimes in the older papers you will see them called oxygen minimum zones, slightly different name for the same thing. They're natural features, so the ocean has almost certainly always had some amount of oxygen deficient zones. They naturally form especially on the west coasts of continents. This is one of the many indirect effects of the Coriolis effect. So because of the direction that the Earth is spinning, you get the accumulation of conditions due to the water currents caused by the Coriolis effect, especially causing upwelling. So specifically the consequence here is that you get a lot of upwelling on the west side of continents. Upwelling is a term for the physics that are created on the west coast of these continents, where you have a lot of nutrient-rich waters actually moving up along the seafloor, climbing along the coast. These waters are deep, and so they've been isolated from the surface ocean for a long time. So they've accumulated nutrients that have not been consumed by biology recently. And so when you have this upwelling of nutrient-rich waters, you can get, for example, really productive fisheries. So that's why the coast of Chile is one of the most productive fisheries, specifically because of the physics of this upwelling. All right. Because you have these really productive waters, of course, you have a lot of fish where you have a lot of phytoplankton, and you have so much production of biomass here that when that biomass is then decomposed, as it inevitably is due to the microbial loop, you get consumption of oxygen. And as we mentioned, if you have enough organic matter, you can actually draw oxygen all the way down to zero. On top of that, this upwelling actually keeps the water kind of at this depth and prevents it from sinking, such that you can really drop the oxygen levels down quite close to zero and keep it that way for a long period of time because of this constant upward force of the upwelling water. So those kind of two things together can cause these oxygen deficient zones to be quite large in terms of depth and also quite large in terms of geography. So along the west coast of South America and the west coast of Africa in particular, you can get oxygen deficient zones that extend for many, many, many kilometers and also many, many meters in depth. The two things together, again, being highly productive waters producing a lot of organic matter, that when they decompose, they will consume oxygen, plus the upward force of the upwelling water kind of keeps that water in place and allows that organic matter to draw down the oxygen down close to zero, or maybe even exactly zero. And of course, when the oxygen goes to zero, you get lots of anaerobic microbial activity, including these things in the nitrogen cycle, nitrification, and NMOX. Another way of talking about how extensive these oxygen deficient zones can be, some estimates have them accounting for up to half of all of the nitrogen loss in the ocean. Nitrogen loss, again, being the production of N2 gas. It's called nitrogen loss because the nitrogen gas actually leaves the ocean and goes into the atmosphere. I said on the previous slide that these oxygen deficient zones are natural. They naturally form for the reasons I explained on the previous slide. On top of that, human activities can make them worse. That is, we are doing things that make the oxygen deficient zones to be larger in terms of depth, larger in terms of geography, and more persistent and more anoxic. Those things include, pollution runoff and also just general global warming actually makes this worse because warmer waters have less ability to dissolve gases, and lots of other kind of indirect, more intricate ways are making it more likely for these oxygen deficient zones to form. and once they form, to get worse and to get bigger. And so here are just several examples. Each one of these graphs is from a different location around the world, and the x-axis in these cases is time. And so you can see that over time in each of these locations, some worse than others. This one is a good example how before there wasn't really much of an oxygen deficient zone. So the colors here are oxygen concentrations. So the blues are low oxygen concentrations. So here the oxygen concentration was low in 1960, but now the oxygen concentration is much lower, and the area of very low oxygen is getting bigger over time. And there's some other examples here as well. It's relatively slow compared to some other things that humans cause, but it's also one of those things where it can change slowly, and then all of a sudden a big change. And there are a handful of reports out there of possible examples of this that you can read about. Okay, that's all I'm going to say for now about the human aspect of this, and now I'm going back to the ecology of it and talking about how denitrification and NMOX interact with each other. So as I mentioned, they have many similarities, but they also have some really important differences. Here are two papers that each did important studies on the relative roles of denitrification versus NMOX in different parts of the ocean. They came to very different answers. One paper said that it was all denitrification, that we don't need to worry about NMOX, and one paper said it was almost all NMOX, and we don't really need to worry about denitrification in terms of understanding the nitrogen cycle of the ocean, and in particular these oxygen deficient zones. So how did these two groups, both of them did good quality science. There was not an issue of one group did bad work. This was an issue of you get different answers in different conditions with different techniques. Before I explain a little bit of why they got different answers, first I want to explain why we even care. So I would understand if you're still not sure why we care, whether denitrification or NMOX is processing more nitrogen in the ocean right now. And the reason is these reasons why they are different from each other. So one reason is that denitrification is heterotrophic and NMOX is autotrophic. So from a pure biological and ecological perspective, it would be really important to understand whether nitrogen removal in the ocean is mostly an autotrophic or mostly a heterotrophic activity, right? That would mean very different things for how the carbon cycle and the nitrogen cycle interact. The second reason is because NMOX is only performed by one weird kind of bacteria, just like a couple of species. And so that would be really important to know. If just a couple of species of weird bacteria are entirely responsible for all of the nitrogen production in the ocean, that would be really important to know about. And thirdly, NMOX again does not produce nitrous oxide as an intermediate. So it would be really important to know how much of the nitrogen removal in the ocean involves nitrous oxide intermediate, again because nitrous oxide is an important greenhouse gas. So we want to know about that. So why do you get different answers with different studies? So again, denitrification is heterotrophic. NMOX is autotrophic. That means NMOX does not really care very much how much organic matter is being produced. It kind of carries on much the same whether there's a lot of fish poop or a lot of decomposing phytoplankton biomass or not. So even in areas of the ocean that are not very productive, you don't have much phytoplankton, you don't have much fish, you can still get NMOX carrying on at very similar rates. So that means that if you happen to do your study in a location and a time where you have a big export of carbon from the surface, then you will probably measure a lot of denitrification because denitrification is stimulated by that activity. NMOX will be relatively constant and could be swamped out in that particular time by the denitrification. Here I noticed that the slide is a little bit cut off, so I'm going to fix the slide here. So in addition to the timing, whether you happen to be doing your study when there's an episode of a big pump of biological carbon, it also depends on your sampling methods. So some times when you sample water from the ocean, You will do it in a way where you collect big particles that are floating or even sinking in the ocean, and some water sampling methods do not capture those particles. And so a very tiny technical thing like that, whether you're including the large particles in your water sample or not, can also affect an answer like this, because again, those large particles are full of organic matter and they will stimulate denitrification. If you don't have large particles, then you are not going to stimulate denitrification, and you will probably measure more anomox compared to denitrification. So this is an example of how these basic ecological and biological differences between these organisms correlates to sampling technical considerations and logistical considerations in how you do your study, and they lead to different answers about these big questions about the marine ecosystem. All right, here's one of the figures I showed in the previous lecture that summarizes a lot of the players in the nitrogen cycle. And so here I just want to highlight where anomox and denitrification are being shown in this figure, in case you want to study this later on your own. And here are some examples of organisms that carry out these processes. And it's also highlighting how both of them are producing N2 gas, but anomox is doing so from ammonia, denitrification only from nitrate and nitrite. Finally, I want to finish the story with a practical social application. So leaving the marine ecosystem behind for now, I'm instead talking about Starfleet. This is a clip or a still from one of the Star Wars movies. Here's Starfleet Academy in the background next to the Golden Gate Bridge. And you may not know that Starfleet Academy is based on this real building in California here. It's actually closer to LA than to the Golden Gate Bridge. But it's a real building, and it actually happens to be a wastewater treatment plant. So perhaps the nicest, prettiest wastewater treatment plant that I know about. And that is my segue to talking about how nitrification and denitrification and anomox are important for understanding wastewater treatment. So here is a figure I found on the internet because I had a hard time finding a really nice figure that shows how this works. But here's your raw sewage. One example, this is not the only way to do it, but one example of how these bacteria can be used to clean up water is, for example, you can have nitrification, the nitrifying bacteria to convert the ammonia to nitrate. And they're going to be using oxygen to do this, so they will also draw down the oxygen, which could make conditions more favorable for denitrifiers, who are, of course, anaerobic. They will use nitrate as their electron acceptor, convert the nitrates to N2, so they will remove nitrogen from the system, and at the same time, they will also remove organic matter from the system. So denitrifiers are super useful because they will remove both the organic matter and the nitrate from your system at the same time, converting the organic matter to CO2. Often they don't succeed in converting all of the organic matter to CO2, so one strategy is to follow this up with another blast of oxygen that will fuel heterotrophic bacteria to consume the rest of the organic matter, converting it to CO2. And so finally, your clean water is now free of organic matter and free of nitrogen. So that's just really quickly how it works in principle. And the final point I want to make here is that a lot of this action actually occurs in biofilms. This is true of wastewater treatment plants, and it's also true of life in general. Most microbial activity actually occurs in biofilms. Biofilms being any kind of surface-attached community. So here are various pictures from one study of biofilms, so communities of microbes that are attached to surfaces in a wastewater treatment plant. And next time we will actually talk more about biofilms and their general importance in ecology. But in this example, I want to highlight these images down here where they have used experimental tricks to color ammonia oxidizers and nitrite oxidizers, which are two different classes of nitrifiers. So these are two different steps in nitrification, converting ammonia to nitrate through nitrite as an intermediate. Those are in green, so you can see those cells living here. Sandwiched underneath them are the nitrite oxidizers. So as you can see here, the ammonia oxidizers are green, and then underneath them are the nitrite oxidizers, and you can see how that would be advantageous because the blue guys are using the waste product of the green guys. So the green guys are producing nitrite, and so then that then flows to the blue cells, who then use that nitrite and convert it to nitrate. And then underneath them, or perhaps neighboring them, are these red cells, which are the Anammox. These are the Planktomycetes bacteria that are consuming both the ammonia and the nitrite to produce nitrate in this wastewater treatment example. So main point I want to highlight here is that this process that is being shown schematically here, a lot of the real biology is actually happening with complex communities of cells that are living with each other. So they're complex multi-species communities, and they're almost always attached to surfaces. Okay, and we'll talk more about surface-attached communities and biofilms next time, and I'll see you then. Bye-bye.